All right, so next we're going to have a couple of presentations from researchers at UT who are working on One Health related projects. So first is going to be Aaron Onufrak, and he is a second-year PhD student in Dr. Danita's lab, who spoke on our panel earlier, and that's in the Department of Entomology and Plant Pathology. He received a Bachelor of Science degree in biology from Slippery Rock University and his Master's in Science in Biological Sciences from Wright State University. His research focuses on characterizing the role of the microbiome in modulating the severity of plant diseases, identifying biological control agents for plant diseases, and population and comparative genomics of fungal plant pathogens. And this will be a recorded presentation, but then he'll be available for a live Q&A afterwards. So I'll hand it over to Alyssa now.
Hi everyone, my name is Aaron Onufrak, and today I'm going to talk to you about a portion of my dissertation research titled Regional Variability in the Phytobiome of Black Walnut and its Potential Role in Regulating the Severity of Thousand Cankers Disease. So to start off, I'd like to define what Thousand Cankers Disease, or TCD, is. So TCD is caused by the fungal pathogen Geosmithia morbida and its insect vector, Phytobiome Juglans, or the walnut twig beetle, with susceptible hosts for TCD including Juglans or walnut trees and Pterocardia or wingnut trees, with the most susceptible host species being the eastern black walnut or Juglans nigra. Now when a walnut twig beetle carrying Geosmithia morbida attacks a susceptible host, what can develop over time is the disease called Thousand Cankers Disease. And if this disease is severe enough, infected trees can experience tree mortality 2 to 3 years following infection. So if you're trying to identify a tree infected with TCD, some signs to look for are entry and exit holes from walnut twig beetles lining the stem and branches of a tree. If you peel back the bark from these entry and exit holes, you might observe walnut twig beetles and their larvae inhabiting their galleries, as well as fungal mycelia from Geosmithia morbida lining these gallery walls. Some symptoms of TCD include cankers formed by Geosmithia morbida that extend from the gallery walls or the gallery margins. And if the disease is severe enough, these cankers can begin to coalesce and girdle the tree. And when this occurs, you might observe external symptoms manifest, such as epicormic branching, leaf flagging, and crown dieback. So currently, Thousand Cankers disease has been detected in 15 US states, 9 of which are present in the western United States outside of the native range of eastern black walnut, highlighted here in green. The other 6 are present in the eastern United States within portions of the native range of eastern black walnut, highlighted here in blue. We also have 4 states where Geosmithia morbida has been detected, highlighted in orange, where, however, in these states, Geosmithia morbida has not been detected with any particular diseased trees, but with alternate insect vectors, such as weevils. So one interesting thing about Thousand Cankers disease is that depending on the portion of the TCD distribution that you are considering, differences in disease severity exist. So in the western United States, outside of the native range of eastern black walnut, we see that diseased trees, the symptoms are more severe and they die 2 to 3 years following infection. However, in the eastern United States and portions of the native range of eastern black walnut, we see 2 different disease outcomes. One, similar to what we see in the western United States, disease symptoms are severe and trees die 2 to 3 years following infection. Alternatively, we can also see that the disease is less severe and trees can recover. And this is where our research comes in. One thing that we would like to understand is why we're seeing these differences in the severity of TCD depending on the region that we're considering. So some hypotheses that we currently have are differences in climate between the 2 regions, as well as potential differences in host genotype. But what I'm going to focus on today are potential differences in the host phytobiome, or the microorganisms that associate with the plant host. And these include bacteria, fungi, viruses, and protists. So these microorganisms can associate with pretty much every portion of a plant, including the leaves or the phyllosphere, the stem or the caulosphere, as well as the below ground portions of the plant, such as the roots, the soil directly adjacent to the roots, which is referred to as the rhizosphere, as well as the bulk soil itself. And the reason that we would expect the phytobiome to have a role in regulating disease severity is because it has well-established roles in stress tolerance, nutrient acquisition, as well as in host defense. And this can be through direct mechanisms, so microorganisms associated with the plant can serve to directly antagonize plant pathogens through mycoparasitism and antibiosis, or through indirect methods such as induced systemic resistance or systemic acquired resistance. And our lab was started to evaluate some of this hypothesis starting back in 2018 when a former postdoc in our lab, Dr. Romina Gazis, characterized the fungal communities of walnut tubular galleries from trees in California and trees in Tennessee using culture-dependent methods. So the way she would do this is she would collect wood samples from trees in both Tennessee and Indiana, or in California. She would then isolate fungi from these wood samples, extract DNA from them and identify them, and use their DNA to identify them. And from this, what she found was, so if we look at this graph here, on the x-axis, we have different OTU identities, or operational taxonomic units. And on the y-axis, we have number of isolates. And we have two different bar colors. So light gray is Tennessee and black is California. And what I'd like to point out from this graph is, one, we're seeing that there are different fungi depending on the region that we're looking at. So different fungi between Tennessee and California. And in particular, we're seeing a large number of trichoderma isolates coming from Tennessee. And this is important and exciting because trichoderma is a known biological control agent used in agriculture. This is because it acts as a mycoparasite, or it parasitizes other fungi. And because of this, Romina went on to evaluate the potential for mycoparasitism in 6 of these trichoderma isolates that she recovered. And the way she did this was through dual plate assays. So the way these work is you inoculate 1/2 of a PDA plate or a potato dextrose agar plate with trichoderma and the other half with Geosmithia morbida. And after a few days, if you see a space between the Geosmithia morbida and trichoderma, it's considered to be an incompatible interaction, and the trichoderma species is considered to be not antagonistic towards Geosmithia morbida. However, if you see that trichoderma has overgrown Geosmithia morbida and has begun to sporulate through the culture, then it's considered to be a compatible interaction, and the trichoderma species is considered to be antagonistic towards Geosmithia morbida. Of the isolates that Romina tested, 6 were identified to be strong antagonists towards Geosmithia morbida. And since this study, we have began to expand upon her work with additional trichoderma isolates from Tennessee, California, and Washington, and also different Geosmithia morbida isolates from across the country. However, what I'm going to be talking to you today is about a different study where we expand upon Romina's culture-dependent work by using culture-independent sequencing. Or in other words, what we've done is we've collected samples from different portions of the plant. We have extracted DNA from these tissue samples of the plant and have sequenced the DNA for amplicons that are related to fungi and bacteria so we can characterize their communities this way. So our goal was to characterize differences in the composition and alpha diversity of the caulosphere or the stem and bulk soil fungal and bacterial communities of eastern black walnut and portions of its native and introduced ranges. And our hypothesis was that fungal and bacterial community composition would differ significantly between the native and introduced ranges. So for our study, we selected 3 different states: Washington, where TCD is present; Tennessee, where TCD is present; and Indiana, where Geosmithia morbida has been detected. The area that we sampled from in Indiana, however, Geosmithia morbida had yet to be detected, so this was an entirely TCD-free area. We selected these 3 states because they had the presence of 4 different walnut clone accessions from the Hardwood Tree Improvement and Regeneration Center. So this would allow us to control for host genotype effect, which is very rare in hardwood tree phytobiome studies. From each of these areas, we collected 3 replicates per genotype, and we also collected wild-type trees as well, with 3 replicates per wild-type trees as well. So for our study workflow, what we had done was we collected bulk soil samples that we extracted DNA from and also characterized soil physical-chemical properties. We also collected branch segments from which we peeled back the bark and collected drill shavings from the margins of cankers. And these drill shavings were used for the identification of TCD-positive trees using molecular probes, as well as for DNA extractions for characterization of the microbiome. As I mentioned, we extracted DNA from both our soil and our drill shaving samples. We amplified that DNA using primers that targeted the 16S region for bacteria and the ITS region for fungi. And we sequenced that DNA using the Illumina MiSeq platform, process those sequences in mother into OTUs, or operational taxonomic units, which are a proxy for species in microbial ecology, and then analyze that data in R. So if we look at my first set of results, on the y-axis, this is for Caulosphere Alpha-Diversity. On the y-axis, we have richness and Shannon diversity, which incorporates both richness and evenness. And on the x-axis, we have our states, so Indiana, Tennessee, and Washington. And we're looking at both fungi and bacteria. And what I would like to point out is that in the native range states, so Indiana and Tennessee, we're seeing higher levels of richness for both fungi and bacteria, as well as higher levels of diversity for both fungi and bacteria, and what we're seeing in the introduced range in the state of Washington. So this suggests that in the native range, we're seeing the native range trees have higher levels of alpha diversity than the introduced range. Next, what I'd like to talk about is the composition of these Caulosphere fungal communities, or these stem fungal communities. And we can do this using principal coordinate analysis, or PCOAs. So if we look at this graph here, points are colored by their state of origin, so Indiana, Tennessee, and Washington, and they are shaped by their clone or host genotype. What we're seeing is that points are largely grouping by their state, but states are largely grouping by region. So our native range states are grouping together, whereas our introduced state of Washington is very dissimilar and further away from these two states. So it looks like the fungal communities of stems from eastern black walnut are more similar to one another in the native range than they are from the introduced range. Additionally, I'd like to point out that we're seeing that clone identity has some role in explaining the variance in these fungal communities. However, it looks like state or region has a much larger role in explaining the variation in fungal communities of eastern black walnut stems. Another analysis that we performed was indicator species analysis. So this would allow us to identify OTUs characteristic of the conditions in certain areas. The areas that we looked at were Indiana and Tennessee, and then TCD positive and TCD negative trees. Now you might be wondering, where is Washington? Well, all of our Washington trees are in this TCD positive and TCD negative tree group. And that's because all of our TCD positive trees were from the state of Washington. So of our 14 trees that we tested for TCD in Washington, all 9 of them tested positive, where the other 5 tested negative, and we had no trees in Indiana or Tennessee that tested positive for TCD. Some interesting indicator O2s that stood out to us were the indicator O2s identified to the Trichomeriaceae, which were present in Indiana and Tennessee plus Tennessee. Fungi from the Trichomeriaceae cause black sooty molds and are common in areas where honeydew is being produced by different insects. So this suggests that the trees in the native range of eastern black walnut are interacting with different insects than they do in the introduced range. Another thing I'd like to point out is that in our TCD positive trees in Washington, some indicator O2s that we're seeing are, first of all, a Geosmithia O2, which is a good sign. We would like to find that in our TCD positive trees, we would like to find our fungal pathogen as an indicator of disease. We're also finding other fungal pathogens such as Alternaria metachromatica and an O2 identified to the fungal family Leptosphaeriaceae, which includes both saprotrophs and fungal plant pathogens. We also characterized differences in the fungal functional guild between each state. So a fungal functional guild can be identified using a database called Funguild. This will give us an idea of the potential functions that a fungus could carry out. So looking at this graph here, we have relative abundance on the y-axis and then state on the x-axis, so Indiana, Tennessee, and Washington. And overall, we're not seeing any major differences between the fungal functional guilds that comprise each state. One thing that did stand out to us was that we see differences in the relative abundance of mycoparasites or fungal parasites. And these are fungi that are antagonistic towards other fungi. And in particular, we're seeing that the native range states, Indiana and Tennessee, have a greater percentage of these O2s identified to fungal parasites than what we're seeing in Washington. We also went a little bit farther and characterized the communities of these fungi identified as microparasites and plant pathogens. And we did this using a principal coordinate analysis, or a PCOA. And as similar to before, points are colored by their state of origin and shaped by their clone identity. And what we're seeing is that for mycoparasites and plant pathogens, communities of these organisms are more similar to one another based on their state and region, so that the native range states have mycoparasite communities that are more similar to one another than the introduced states. So this suggests that Geosmithia morbida is interacting with different mycoparasites and plant pathogens depending on the portion of its distribution that it's in. Next, we went on to the Caulosphere bacterial communities. So similar to what we saw with fungi, overall community composition is more similar in the native range states. So once again, this is a PCOA with points colored by their state and shaped by their host genotype. We're seeing that Indiana and Tennessee are more similar to one another than they are to Washington. However, this time we're seeing that host genotype does not have a strong role or does not have a significant role in explaining the variation in community composition. However, we are seeing that region once again is explaining a significant portion of the variance in these bacterial communities. When we consider the indicator species, using our indicator species analysis, once again, we have Indiana, Tennessee, and then our TCD positive trees in Washington and our TCD positive trees and our TCD negative trees in Washington. What we see is that for our native range trees, one OTU that's sticking out is the Spirosoma species. We also have this in our TCD negative trees. So Spirosoma is a common endophyte that is recovered from many plants. However, its role in the microbiome is not well understood. So that's one avenue of research that we need to continue and follow up on. Interestingly, in the TCD positive trees, we found bacterial species identified as diazotrophs. So diazotrophs are nitrogen-fixing bacteria. So one potential reason that we're seeing these diazotrophic bacteria in our TCD positive trees is one, all of these trees were located in Washington, where soil conditions, nitrogen availability in the soil was considerably lower than what it was in the native range soils. So these nitrogen-fixing bacteria may be helping the plant host acquire nitrogen. Alternatively, it's possible that Geosmithia morbida alters nitrogen availability following infection, and so by fixing their own nitrogen, these bacteria are able to thrive in the stems of these host trees. So that's everything that we have for the Caulosphere. So this brings me to the conclusions for the Caulosphere. So first, what we found is that there's lower levels of richness and diversity in the introduced range, and this could be a product of just being sick with the disease. So maybe these infected host trees are not able to support as large of a diverse community as healthy trees. Alternatively, it's possible that these higher levels of richness and diversity in the native range may serve to limit the establishment success of Geosmithia morbida through competitive exclusion. And then lastly, we see that the native range fungal communities contain a greater proportion of potential mycoparasites. And these could serve as novel enemies for Geosmithia morbida, particularly because we know that the identities of these the community composition of these mycoparasites differ between the native range of eastern black walnut and the introduced range of eastern black walnut. When we move into the soils, so we'll start off with soil alpha diversity, similar as to what we did in the stem. What we're seeing is a little bit different this time. So on the y-axis, once again, we have richness and Shannon diversity. And on the x-axis, we have our states, so Indiana, Tennessee, and Washington. And we have fungi and bacteria. So for fungi, what we're seeing is that similar to the Caulosphere or the stem, we see that in the native range trees, alpha diversity or richness is higher than what we're seeing in the introduced range of Washington, but it's flipped for bacteria so that in the Indiana and Tennessee, we're seeing lower levels of alpha diversity in the native range than what we see in the introduced range for both richness and diversity. When we consider the soil community composition, this is similar to what we saw for fungi. Once again, these are PCOAs, or principal coordinate analyses. Points are colored by their state of origin and shaped by their host genotype. And what we're seeing is that our native range states, so Indiana and Tennessee, are more similar in composition to one another than they are to the introduced range state of Washington. However, there's considerably more overlap in the soil than what we saw in the Caulosphere. Once again, we also see that region is a much larger explanatory variable in terms of the variability in the composition of these fungal and bacterial communities in the soil compared to host genotype. And then when we consider the fungal functional guilds that comprise these soil fungal communities, overall, similar to what we saw in the caulosphere, we're not really seeing any significant differences in the fungal functional guilds that comprise these communities. However, we are seeing differences in the composition of these different fungal functional guild communities. So for instance, arbuscular mycorrhizae, which are used, which associate with plant roots and help them acquire limiting nutrients such as nitrogen and phosphorus, we see that once again, their communities differ between the native and introduced ranges. We also see that mycoparasites differ between the native and introduced ranges, as well as plant pathogens. And so that's it for the soil. So overall, what we're seeing is higher bacterial richness and diversity in Washington soils could be related to higher soil pH. So this is a principal coordinate analysis. And once again, these points are colored by state. And what we see is that in our native range states, so Indiana and Tennessee, these soils have lower pH values than our introduced range state of Washington. And we know that for microbes such as bacteria, pH tends to be a really strong determinant of species richness, typically with increasing pH leading to increased bacterial species richness. These differences in the community composition are likely related to differences in the soil physicochemical properties between these three regions. And finally, the available species pool in soils for eastern black walnut to associate with differs between the native and introduced ranges. And this is probably the most important thing from this because we know that phytobiomes are nested. And that nesting starts within the soil and then works its way up to the leaves. So this suggests that because the species in the soil are so different in each region, the species available for the plant to associate with are going to differ. And this could have impacts on the host's health. So the overall big picture is that the composition of the J. nigra phytobiome differs between its native and introduced ranges. We see that there's greater fungal and bacterial diversity in the native range, which could contribute to disease resistance or could be a product of disease. And we also see that the composition of mutualistic and plant pathogenic fungal functional guilds differs between the native and introduced ranges. This is true for both above ground portions and below ground portions of the plant. So where do we go from here? So as I mentioned earlier, all of our TCD positive trees were in Washington. So this makes it difficult for us to disentangle the effects of disease versus the effects of just being of geography. So what we wanted to do was to assess what is just the effect of hosts being infected with 1000 Cankers disease on the microbiome, as well as the host gene expression. So this past summer, we inoculated 72 trees with Geosmithium morbida, and we collected tissue samples from areas near the margins of these cankers, from which we would extract DNA and RNA. And this will allow us to characterize the effects of TCD on both the host microbiome as well as host gene expression. Additionally, we wanted to begin to evaluate potential biological and chemical controls for 1000 Cankers disease. And one thing we wanted to evaluate was the effects of these chemical and biological controls for TCD on the host microbiome and gene transcription as well. So in this study, we also treated these trees with a chemical control for TCD and a potential biological control for TCD as well to see their effects on the host microbiome and host transcriptome. So with that, I'd like to acknowledge my committee, as well as Jeffrey Williams, who was who co-wrote this manuscript with me. Jeff Williams is a PhD student out of Dr. Mackinzell's lab in Purdue, and he was a pleasure to work with. I'd also like to thank my advisor, Dr. Janita Hadjiabdic, and the rest of my committee, as well as all of my lab members and colleagues who helped me with this research. I'd also like to thank all of our funding institutions, as well as the USDA, the University of Tennessee Institute of Agriculture, and the Entomology and Plant Pathology Department here at UT. And with that, I'll take any questions.
Okay, thank you, Aaron. And Aaron is here with us live. If you guys have any questions for him, you can type them in the chat or also use the raise hand function to be unmuted.
Hi. Hi, Erin. I'm sorry, I cannot find the raised hand function. I don't know if anyone else is having issues with that, but I guess I'll just ask one question to get started. So I actually forgot what I was going to ask. I got confused about the raised hand function. It's under participants. Gotcha. Thank you. Crap. Oh, the pH. So, you mentioned that's kind of a really interesting concept to have such a relationship with bacterial richness and pH. I'm assuming there's at least a threshold in the soil where, I mean, if I throw a bunch of lime in the soil, I'm not going to get extra species. Is there any kind of cutoff where that relationship ends, just out of curiosity?
Not that I'm aware of. There's probably a point where, like, you get too acidic and it's not going to grow, or too basic and you're not going to get bacteria, or you're going to get certain types. But I'm not sure that there is an actual like threshold where it stops changing.
Right, well, I'll just keep one more question going then. It's just okay with everyone. So with the with kind of looking at maybe certain bacterial species that prevent or compete or actually actively kill this pathogen, is there any research or something you're looking at where maybe by itself a particular bacterial species isn't going to be that, kind of antifungal, but if it's in a community with other species where it gets signals from a group of bacterial species, and as a group, they're kind of a stronger antifungal kind of cohort?
Right, we haven't tried anything like that. One thing we've, coming from like the majority of our culture work with these, with just morbid has been with fungal pathogens or fungal microparasites, and looking into that avenue. But definitely one thing that we would like to do is do both redo the culture-dependent study with fungi, but also with bacteria, so that we could then go forward to see, well, if we change the levels of richness that we and the number of species that we challenge geosmithy with at a time, so more than one species are increasing, sort of like you said, do we start to see differences in the efficacy of controlling this pathogen? And that's something that's totally possible. It's just something we haven't gotten to do yet, but something we'd like to do down the line.